please consider joining. So I'm pleased to turn over the podium to today's panel organizer, Dr. Samantha. I'm pleased to say Dr. Samantha as well, because Dr. Samantha Yammine finished her PhD, she just told me, in April, which is great. She joined our program committee a year ago, and we are delighted that she's helping create some of our programming. The program committee, for those of you who don't know, of RCI Science is the really fun committee where we decide what kinds of events we put on. And Sam has come up with some fantastic ones. She is a neuroscientist, science communicator, and digital media producer. She got her PhD from the University of Toronto, researching how stem cells build the mammalian brain before birth and maintain it throughout adulthood. Sam is passionate about empowering people to explore science by making it more familiar, accessible, and inclusive. She's a frequent guest on CBC Radio, you may have heard her there, and she's worked with ASAP Science, TVO Kids, Leafs Nation Network, 3M, all sorts of things. You can learn more about her at her website, which is heysciencesam.com. And with that, I will turn it over.
Well, hello everyone. Sorry, let's do that. I'm delighted to be here today and so wonderful to see so many faces in the audience, excited to learn about this topic that is near and dear to my heart. I'm also very grateful to be joined by an esteemed panel of fantastic experts in the field of stem cells and regenerative medicine, and experts all across the spectrum from research all the way to figuring out how we can implement that research and get it into patients when it's ready. So joined here today, we have Dr. Nika Shakiba. Yes. Yes, I like this energy. Let's do that for everyone. We have Dr. Lise Muncy in the middle. Round of applause. Fantastic. And furthest from me, we have Dr. Shifra McMahon. I'm going to give you, they all have such fascinating bios that really contextualize why we wanted to have them up in the panel. But first, I just wanted to say a few words. This topic of stem cells in regenerative medicine, as someone who's been researching stem cell biology for about 7 years now, I've become so fascinated and learned so many fantastic things about the field of stem cell biology that have made me really hopeful for the hope that it can have for people around the world at all sorts of ages dealing with various different conditions. I'm also fascinated by what stem cells can teach us about our bodies and how they form in development. And we're going to hear a lot about all of these different sides and insights that stem cell research brings us from some of our panelists today. But part of my job is also communicating what we know about stem cells and what we don't know. And I started that a few years ago because the more I learned about the technical side of stem cell research, the more I also saw it being exploited in the popular media. And I saw, it's a field that's rife with celebrity endorsements and people taking advantage of vulnerable patient populations and preying on their fear with promises that are just not substantiated. And so our goal, everyone at RCI Science who was excited about this topic, and the people who joined me up on stage today, our goal is that all of you tuning in virtually or here in person are going to feel empowered to be as hopeful and as excited as we are about stem cells, but to be carefully critical as well, and to become ambassadors for more truth-telling about stem cell research going forward, starting from here today. So how this is going to run is going to introduce each of our speakers, and one at a time, they're going to come and give you a little overview of their piece of the pie and their little expertise in stem cells. And then we're going to have a moderated Q&A. I'm going to ask them some questions to get us going, but then I'm really going to be looking to you to ask brief but important questions that maybe you've been thinking about for a while or that pop up throughout the session. So if you have any questions as we go, please jot them down and hold them towards the end, and I promise that we'll have a really interesting Q&A period. So with that, I would like to introduce our first panelist, who is a longtime friend of mine, Dr. Nika Shakiba. While Nika is now a postdoctoral fellow at MIT, she began her stem cell research really close to here, actually, just in the adjacent building, on the U of T campus as a bioengineer in the world-renowned lab of Dr. Peter Zandstra. In his lab, she studied the process of reprogramming skin cells into stem cells, which we'll hear more about later. And now at MIT, she studies synthetic biology to engineer stem cell behavior, an exciting new field that we'll start hearing more and more about in the headlines, I'm sure, in the coming years, thanks to some of Nika's work, too. Nika is a leader in outreach and mentorship. She's the poster child of many things at U of T now, even though she's gone. But we're lucky to have her here with us today. She's flown up from Boston, and I'm so excited to hear what you have to share with us, Nika. So come on over.
Thanks so much for that intro, Sam. It was really my pleasure to be here today, and I'm hoping that after my talk you can have as much excitement as Sam had said about stem cells as we do working in the field. And so my job here really is to introduce you to some of the breakthroughs, but also some of the unknowns that remain in the stem cell field. So I'm going to take a step back and make sure that we're all on the same page in terms of what are stem cells, how do we define them, where can we find them. And the way I really like to think about the potential of stem cells is in the context of human development. So if we think back to when we first began life, I mean, you probably can't remember the time that you started life, right? But it goes all the way back to when the fertilized egg formed. So when the sperm and the egg meet, they form this fertilized egg. And that one single cell, that is a stem cell, goes on to divide and reorganize itself and produce this magnificent, complex being that is the human body. And that whole human body, all of those different cell types, started from one single cell. That's a truly magical event, right? And that is really the power of a stem cell. So what does a stem cell mean? How do we define stem cells? So there's really two key properties that a stem cell has. The first is that it's able to self-renew, meaning that it can divide and make copies of itself, theoretically infinitely many times. And the second property is that while a stem cell itself is not specialized, it doesn't beat like a heart cell, it doesn't spread out and cover our body like a skin cell, it's able to give rise to all of these specialized cell types that allow us to function. So it undergoes differentiation to perform all these specialized cell types. But not all stem cells are the same. So there are different classes and types of stem cells. So I mentioned the fertilized egg, the zygote. That single cell, that is considered a totipotent stem cell. Toti meaning that it has total potential. So it has the ability to give rise to all of the cell types in our body, but then also the extra embryonic tissue that supports our growth as a fetus. So umbilical cord, placenta, that comes from that totipotent stem cell. But as that totipotent stem cell is starting to divide and start to form the embryo, the blastocyst is the next image I'm showing there. That blastocyst has already started to specialize some of its functions. The cells on the outside are forming the extra embryonic tissue, but the cells on the inside, or the inner cell mass, go on to produce our body. So the cells on the inside of the embryo are a type of stem cell we call pluripotent, pluri meaning many. It has much potential. And the potential it has is really to produce any of the cell types in our body, no longer the umbilical cord and placenta, but any cell type in our body. And then that embryo goes on to divide further, and the stem cells specialize further, and we get what are called multipotent stem cells. They do have multiple potentials, but their potential is now always restricted to the organ or tissue that they're in. So as our adult body has formed, we still have these stem cells in our body. They're multipotent stem cells. We call them adult stem cells, and their job is to maintain the tissue or organ that they're in. So for example, our brain has brain stem cells, neural stem cells. Our blood has hematopoietic stem cells. They can produce all of the cell types in that tissue and organ, but they can't produce cell types from other tissues and organs. And then these multipotent stem cells, as I mentioned, go on to actually produce the functional specialized cell types in our body. And for many years, scientists thought that this was a unidirectional trajectory. Once a stem cell has, differentiated and made itself into a skin cell, it's stuck there. It's not going to change into a different type of cell. But recently, stem cells have started to think about, what makes a cell a cell? What defines the identity of a cell? And can we somehow manipulate the identity of a cell? And maybe take a skin cell, for example, that's fully specialized, and convince it to go back and become a stem cell again. Can it regain potential? Can we reprogram it? And in fact, in 2006, it was shown that we can do this very thing. We can actually take skin cells from anyone, really, and we can genetically engineer them so that they basically forget that they're a skin cell, and they go back to this unspecialized state. They become very much like the pluripotent stem cells you'd find in the embryo, which are often called embryonic stem cells. And so we've really induced the pluripotency in these cells, and therefore they're called induced pluripotent stem cells, or iPS cells. And so there's a lot of opportunity here, right? Because I can take my skin cells, turn them into stem cells, and then I can use those stem cells to produce any cell type in the body. Why not? I know I can make organs for myself, theoretically. And so this idea of reprogramming has really sparked a lot of excitement in the field and outside of the field, certainly. And this was the fastest turnaround time for a Nobel Prize to date. So 2006 discovery, 2012 Nobel Prize. So it's a really kind of powerful idea. And I'll talk about iPS cells in the context of some other sort of recent exciting research as well. But in general, the idea of stem cells, whether we find them in our body, whether we produce them in the dish, they really are the cornerstone for regenerative medicine. So maybe you've heard of regenerative medicine, the idea that we can kind of regenerate function in our bodies. And there's kind of different ways that stem cells can have a role to play in that. One very obvious one is one I mentioned, right? Like we can take stem cells, produce different cell types, maybe we can make tissues and organs, put them back in the body. We won't need donors for organs anymore. And that's the idea of cell and tissue therapy there I've listed on the bottom. But there's other things we can do too. If we know that our bodies have adult stem cells, what if we could learn how those adult stem cells work? What if we could better communicate with those adult stem cells and convince them to repair our bodies within the body without, having to take cells out, manipulate them, and put them back in? Maybe we can just give a drug that stimulates our endogenous adult stem cells to do their job a little bit faster, and maybe we can replenish some of our lost function that way. And for me, I am an engineer, but I'm a basic biologist in a lot of ways. I'm really interested in understanding how is it that this one cell goes on to form this magnificent body. I mean, not that my body's that magnificent, but our magnificent bodies, right? Like, how does that process happen? It's truly magical. So that's really, the understanding the biology of tissue development is what really excites me. But so I wanted to give some examples of these, and one of the, most well-known, I guess, examples of what a stem cell might look like as a therapy is a bone marrow transplant. So who's heard of bone marrow transplants? I mean, I hope we all have, because, Canadian Blood Service is always asking us to sign up to, be a bone marrow donor. But bone marrow transplants are really the earliest example of a stem cell therapy, and probably one of the most, pronounced popular ones, and certainly we'll learn more about ones that are kind of trickling down the clinical trial pipeline, but this is one that's established and we've been using since the 1950s. So what's really the magic sauce in a bone marrow transplant? I don't think many people knew. I certainly didn't know before I started my PhD, but that is really the blood stem cell, the hematopoietic stem cell. So that blood stem cell, as I showed, sorry, let me go back here. This blood stem cell sitting at the top of this hierarchy can produce all of the blood cell types in our body, right? So every red blood cell, every white blood cell, your immune system, all comes from this one blood stem cell. And so it's quite powerful, right? So it can be used to treat a lot of things, and it has been used to treat leukemia, lymphoma. These are very sort of typical diseases that we'll treat it with. And we extract the bone marrow from, the marrow, right? So usually we go into your hip bone or your femur, and we'll extract it. And these cells can then be, transplanted into a patient that needs them, and it'll replenish their whole blood supply. So it's quite powerful. It's a very widely used cell therapy, but we don't have that many donors. We don't have that many matches. There's not enough cells. So there's still a lot of questions there. But I love this example of the bone marrow transplant because it really brings us back to our roots as Canadians. Not many people knew that the first stem cells ever were discovered in Canada, just down the street. And as I was bringing my aunt and my mom over, we actually stopped at this very statue that recognizes Drs. Till and McCullough, who were, the grandfather of stem cells. And we took a little photo. I encourage you all to walk back that way past the Mars Center. You'll see their statues there. But they discovered the blood stem cell. So, we as Canadians have roots in the stem cell field. So I'm really excited that we can kind of carry this legacy forward. So I also want to talk about some examples about things that we're working on. Not quite there yet, but the idea that you could take, skin cells, reprogram them, make them into iPS cells, and then use those iPS cells to make any cell or tissue type in the body. we can't really do, full organs yet, but we've gotten to the point where hopefully, I don't know if the video is playing. Oh, maybe we'll click on it. Oh no, is that not working? Well, anyways, that's okay. We really hyped this video up, but essentially, what apologies. I hope we're all awake now. Wait, there must be a way to mute this video. Yeah. There we go. Surprise! Yeah, they are amazing, just the way they are. So these are heart cells that we've derived from stem cells in the lab. And you can appreciate that's not a full heart, right? But we can get them to twitch. They kind of make these balls of cells that become heart cells, and they can twitch, and they have some function of heart. But they're not quite the heart itself, right? So we are making some kind of inroads into developing different tissues and organs from stem cells. And the vision, certainly, if we can get rid of this, the vision certainly is to, produce various cell types and inject them back into the body and hopefully, repair someone's damaged heart after a heart attack. We're not there yet, as I mentioned many times, but we're getting there. And that's kind of one of the visions of cell and tissue therapy from stem cells. The other one I mentioned is the idea that we can kind of speak to the endogenous stem cells that we have in our body's adult stem cells, and convince them to work faster, divide faster, make more copies of themselves so that we can replenish tissue that's lost. For example, after a stroke, there's damaged brain tissue. Maybe, we can derive drugs that can stimulate those endogenous brain stem cells to start dividing and differentiating to repair some of that lost brain. These are kind of big picture ideas that we as scientists are working on, but that's kind of the vision of what a stem cell could be powerful and used for. Other things that I find interesting really is the idea that we could take stem cells out, grow them in the dish, and follow them along as they're producing various cell types so we can understand how our body does this during embryonic development. That's fascinating to me. And certainly more closer to home, the idea that you could, instead of, convincing a Parkinson's patient to give me a slice of your brain, instead, I could maybe just take a cheek swab and use the, skin cells that we get from that cheek swab, convert them to stem cells, and then turn those stem cells into brain cells. Now I have an image of that person's brain cells without cutting into their brain, right? And so I have a Parkinson's patient cells that I can use to study drugs. I can use to study the process of Parkinson's. How does it come to be? How can we intervene and help it? These are the things that sort of reprogramming iPS cells and the field of stem cell research has been working on, and some really exciting sort of cutting edge things. So what have we learned in the field? Certainly that stem cell exists, that we can use them to kind of program them to make different cell types on demand, that we can reprogram cells, turn cells that weren't stem cells back into stem cells and then use those cells for regenerative medicine purposes. But of course, we're aware of the things we're not yet able to do completely. We can't really stimulate our endogenous stem cells well yet. We can't produce 3D organs on demand, although there are some interesting technologies that are emerging, and I'm happy to chat about further. If not, our other panelists will talk about. So we're getting there, but we're not quite there. But, it's a very exciting time. And we as Canadians should be proud that the legacy started here, and we should all kind of be working together to push it forward as much as we can. Right, with that, I'm to the end of my talk. Thanks so much for your time and attention.
Thank you, Nika. And I apologize about the sound. See, the backstory is that Nika checked in with me like 5 times because Nika's very organized. And she's like, my video works, right? The sound won't play, right? And then, of course, I said, yeah, no problem. Got it. But surprise again. That shouldn't happen again, but who knows? You never know what happens at an RCI event. Okay, so our next 2 speakers, I want to give a few words on where they work. They're both from the Center for Commercialization of Regenerative Medicine, which you'll probably hear us refer to as CCRM for short, because it's quite a mouthful. And I want to just give a few words on what CCRM does, because it's very unique, it's very fascinating, and we're very lucky to have it here in Canada. It's a not-for-profit, public-private consortium. And the reason why it's important to have this kind of thing here, where there's so much legacy with stem cell research, is that many new and potentially life-changing regenerative medicine-based treatments never reach patients because they're not successfully moved from the laboratory to the stage where they can actually be used in medicine. In order to fulfill regenerative medicine's promise to treat the many diseases affecting our population, a world-renowned group of stem cell scientists and bioengineers came together to form CCRM not too long ago. And now it continues on its mission to generate sustainable health and economic benefits through global collaboration in cell and gene therapy and regenerative medicine. So it's a very, they perhaps can talk a little bit more about it, but it's a very unique organization, and so there's a lot of expertise all across the research pipeline, and our two speakers today will be good evidence to that. So first, we're gonna hear from Dr. Lise Muncy, who is a Senior Development Manager at CCRM. Dr. Muncy received her PhD at McMaster University, focusing on drug discovery efforts in neurodegenerative diseases, specifically Huntington's disease. Following this, she held a postdoctoral research fellowship at the University of British Columbia, where her studies focused on the genetic causes of Parkinson's disease. When she's not working on the front lines of taking regenerative medicine beyond the lab bench, Lise is also an incredible and adventurous athlete, so she has some cool stories if you want to chat with her after. So without further ado, Lise, we'd love to have you up here.
Hi, everyone. Thanks for coming, and thank you, Sam, for that intro, and thank you, Nika. That was a really great kind of intro into stem cells, and I hope it's really kind of sparked your imagination to the tremendous potential that stem cells truly have to change the face of healthcare. As Sam mentioned, myself and Shifra work at CCRM, and our mandate is facilitating moving the most promising regenerative medicine therapies from the lab bench into the clinic. So I've titled my talk, and I was asked to talk about the future of stem cells, and the reason I have that in quotes is because truly the future is now. Everything I'm going to talk to you about may kind of sound like science fiction, but it's things that's happening across the world and in Toronto as we speak. So as Nika mentioned, stem cells are these powerful little cells that have the ability to turn into any cell type in the body. So for instance, heart, brain, or liver cells. So scientists have spent the past decade figuring out what environments to put them in and what signals to turn them into their cell type of interest. So let's take brain cells, for example. So brain cells are called neurons, and your brain is made up of about 1 billion neurons, but not all neurons are alike. Of course, on top of that, there's about 100 subtypes of different neurons that make up your brain. So traditionally, what scientists have done is made one kind of neuron and in a 2D dish. So as much as this is amazing and we can look at the cells and we can ask some questions, we don't always get the answers we expect. So say in this example, drug A is meant to elicit a response in the brain, we might not see this on the dish. And the reason is because that blue brain cell normally talks to the yellow or the purple brain cells. They grow in 3D in the organ in the brain and they talk to each other. So scientists recently have gotten super creative, and they've basically made these gels that they inject the cells into. So now the cells are growing in 3D in an environment that's very similar to the organ they came from. So in this example, the brain. With the right signals, these cells will turn into the different subtypes, and they'll actually start talking to each other, and they'll actually form a little tiny organ. Delightfully, scientists have named these organoids. And generally, if you put drugs on those, they do elicit a response very similar to what you would see in the brain. So this is what is being used now to expedite drug discovery, as well as potentially be used in transplantation medicine in the future. So another really hot topic is called personalized medicine. Now, this can mean a lot of different things. The context I'm going to be talking to you about has to do a lot with stem cells and using your cells both to discover drugs that are specific to you or to make cells that are transplantable for yourself. So I think most people here probably know that every cell is made up of DNA. That's our genetic blueprint. And this blueprint is 3 billion different little codes. So every single person, if you look at the person next to you, that coding is 99.9% the same. So that 0.1% is 3 million differences. I'm looking at my brother, he's in the front row. Does that math seem about right? So you have about 3 million different base pair differences from the person next to you. And that's what leads to our diversity, which is really great. However, that also is why a drug may act differently in me than it does in someone else. And that's also why we can't necessarily use cells from one person to treat another. So a lot of diseases are now known to be caused by one change in that 3 billion code. So lots of the different work I did in Parkinson's disease, for an instance, we would find one difference in your genetic code, and that would cause you to have Parkinson's disease. So those cells, our own cells, may not be good enough to treat ourselves. So can we use a cell from another person? And the answer is not always yes. And again, this is because of these very subtle changes in the DNA. But of course, scientists have found a way to deal with this. Now, probably a lot of people, and that again is why you can't necessarily use someone else's cells for transplant. These different little genetic codes mean your body recognizes as not yourself, and I'll get into that in a little bit. So again, something that's been a lot in the media is this term CRISPR. And what CRISPR is these genetic scissors that we can actually go in as scientists. We can go into the stem cell, we can find that disease-causing mutation, and we can cut it out, and we can replace it with DNA that doesn't harbor that disease. So if you carry one of these mutations, we can make stem cells that no longer have the disease, but the rest of the genetic background is identical. We can then, as Nika mentioned, make an indefinite amount of these cells. We can do drug discovery on them. And then, of course, in the future, these would be disease-free cells that won't cause rejection in yourself. So this is what personalized medicine means. Now, why are these cell therapies not getting to the clinic? You know, we talk so much about them, and a huge part of this is time and money. So the technology Nika was talking about, where you take a blood sample and you turn it into induced pluripotent stem cells, with current technology and doing this in a clinical grade manner, this is going to take up to 6 months. So that's 6 months of someone working to get your blood cells or your skin cells into stem cells. Then following that, you have to grow about 1 billion of them to make something that would be therapeutic. Up until a couple years ago, we couldn't do this. We could not grow this many stem cells in one go. Recent technology in bioreactors, so basically growing stem cells in big vats, is brand new in the past couple years, and we can now routinely do this. So we've kind of tackled that. However, then you have all these stem cells, and now you need to differentiate them into whatever cell type of interest. Again, pancreas, lung, heart, brain. That takes another 1 to 8 weeks, maybe even longer. So you're talking, up to a year, and I don't know why that. Anyway, that should say cost of $1 to $2 million. So that's an autologous stem cell therapy. So again, these costs anywhere from, there's the 2 million, $1 to $2 million for one therapy for one person. So what autologous means is putting your own cells back in your own body. Now, if we could just make one cell line and treat many people, this would cut the cost significantly. So if you could take someone's cells, make stem cells, and then make 40, 50, 100 billion of them, they turn into all the different cell types, and you could treat hundreds of different people. We call this allogeneic. So an allogeneic therapy is where we're trying to move, but again, the problem with this is rejection. Your body may not accept cells from somebody else. But of course, scientists are tackling this. So the last thing I'm going to talk to you about is this new technology called cloaking. So part of what happens is these 3 million different changes in your cells versus the person next to you make these little proteins on the surface of your cells, those little green guys. So what that means is your body knows those are yours, but they tell everyone else that it's not theirs. So we have an immune system, these things called T cells, and they recognize those proteins. And if they're not normal to you, they attack them, and that's what causes rejection. So that's the same way it would notice a bacteria or virus in your system is not yours. It would recognize someone else's cells. So scientists have looked into that 3 billion code of DNA, and they've actually found the parts of that code that encodes those proteins on the surface. So they can go in with their genetic scissors, and they can cut those proteins out. So that's not happening anymore. Now that cell is cloaked. Think of an invisibility cloak from Harry Potter. You put that cell in your body and the immune system can't see it anymore. So these are kind of able to be, again, could be from one person to the other. And now we would have our cost-effective allogeneic stem cells. If we can do this, that would mean we can have an unlimited source, what we would call a universal stem cell. Now, this is, again, an active area of research. It's not in the clinic yet. Researchers are still trying to figure out how safe it is to cut up this DNA and then put cells back into a person. And of course, there's ethics around, is it okay for us to change our DNA and put these cells into a person? So that's kind of where I'm going to end my talk. I'm really excited to answer any questions you guys have about these technologies or any other questions you have about stem cells. And I know my colleague Shifra is going to talk to you about the current landscape of kind of where we're at with the clinic. So I hope you're all really going to enjoy her presentation. Thank you.
All right, our final speaker before we head into our Q&A is the wonderful Shifra McMahon. Shifra is the Senior Manager of Clinical Translation and Regulatory Affairs at CCRM. She works with academic and industry partners to advance promising products and technologies to commercialization so we can actually get them out there. Having worked worldwide, Shifra brings more than a decade of experience in pharmaceutical and biotech regulatory affairs and policy. She has some fascinating insights to some of the regulations and ethics surrounding stem cell therapies. And truly, I could not be more excited to pick your brain because she's actually out there doing all this stuff. If you're wondering, why can't I go inject stem cells here and be cured and blah, 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 she will tell you why. So come on up, Shifra. We can't wait to hear from you.
Thank you. Hi, everybody. It's great to be here. I will just do a little correction of an intro earlier on. I am not a PhD, so I'm gonna remove that doctor from the original intro. No, that's fine. Not a problem at all. I just didn't want to accidentally take it. So before we go any further, quick show of hands. Who here knows what regulatory affairs is, or maybe knows somebody who works in it, or has had some experience with it? Okay, some hands, that's good. Sometimes there's no hands, so that's excellent. I'm going to talk a little bit about what we actually mean by regulatory affairs in the broader sense, so that we've got a bit of a baseline for what we're going to talk about afterwards. So, broadly speaking, on one side you have government, which has as an aim to protect us as a general public, so to ensure public health. And when it comes to drugs and devices, pesticides, agrochemicals, all these things out there that have potential harm, governments are looking for evidence of quality, of safety, and of efficacy for these products. On the other side of the bridge, you have companies who are developing new technologies, new drugs, new devices, new ideas, and their aim is to bring these to the market and to sell them. They are commercial organizations. So regulatory affairs does a little bit of a bridging between those two groups. On the one hand, ensuring that government regulations are met, that we are achieving the levels of safety and quality that we expect to achieve, and that we're getting the type of efficacy that we want to get out of these products. And on the other hand, from the company perspective, regulatory affairs professionals are also working to ensure that the entire development of the program and of these technologies is done in line with those regulations. So if there's something that comes up that is just impossible, there's a meeting of minds really to talk about that and to say, this is a brand new idea, it's never been done before, there's no clear regulation on how to do it, but we as a company have this great idea, how do we make this work? And that's really the most important part of regulatory affairs, and particularly in this space, in stem cell therapies, because as you heard from our previous presenters, this is very new. Like the future is now, but we weren't necessarily prepared for it to be today. We were kind of prepared for it to be soon, but we're not quite there yet. So there's still a lot of discussion to happen in this space. So if we look at specifically regulating stem cells, in most countries, including in Canada, they are regulated in the same way as a drug. In fact, they are defined as a drug under our regulations. And there are very good reasons for this. If you are a drug and you're trying to bring this drug to market, you are required to have rigorous clinical trial data. Clinical trials are where we test new drugs in ever-increasing groups of patients so that we don't expose people to undue harm too early. When we don't know yet how a drug works, we expose a very, very small group of patients, or sometimes healthy volunteers, and we watch very carefully to look for safety and to see some signs of efficacy. If that looks like it's acceptable and we haven't harmed anybody and we think we're getting something out of it, we gradually increase that patient population while also starting to look at much more detailed aspects of the drug itself. So as we get later on in that clinical trial program, we're looking for very specific measures of efficacy. We want to be certain that this particular therapy designed to treat Parkinson's is actually going to treat Parkinson's and is not going to do something else or have undue side effects that we can't manage. So a rigorous clinical trial program is important, and it's staged and it's phased, but this is a mandatory step for drugs, and at this point also a mandatory step for cell therapies. The other piece that comes with being regulated as a drug is a significant amount of control around the manufacturing and the distribution of these products. If you imagine a drug, an Advil that you might buy off the shelf in your local pharmacy, those drugs have been produced to a minimum standard, which I should say is a very, very high standard, a minimum standard of quality. That means that the manufacturing site where it's made is regularly inspected. They are required to meet certain standards that ensure our safety, that ensure that those drugs are not contaminated with rat poison or environmental contaminants or somebody who's sick who breathed over the batch and now you're going to get sick because the bacteria is still there. So we're very, very careful to monitor quality of these products. And as you can imagine, with cells, which are living organisms on their own, that level of quality is even more important. We need to be certain that those cells are not active in some way that they shouldn't be, and that they're not contaminated with something that they shouldn't be. Finally, once you've actually gone through all that and you have your clinical trial data and you have good quality manufacturing data, you then want to access the market. The market that you can access is limited and controlled based on the data that you've generated. So if you ran all your clinical trials in Parkinson's disease and you now have a drug that's of high quality that you can demonstrate is likely to treat Parkinson's disease, you can't sell it in Huntington's disease. You can't choose to just kind of go a different way with it. You have to generate all that data again if that's what you want to do. And this is a really important concept to understand because at every single stage of development and every stage of research, we're controlling risk. We're trying to reduce the level of risk that people are ultimately exposed to. And as you can imagine, these are carefully regulated for really good reason. We heard from these guys that you can have totipotent stem cells, multipotent stem cells, pluripotent stem cells. What happens if a stem cell that is supposed to be a neuron in your brain ends up differentiating in your body into a piece of bone? How do we manage that? And these things are extremely important. That's in a very tightly regulated space where they're clearly drugs. Unfortunately, what we're seeing is a lot of people who talk about using stem cells in spaces where they're not really drugs, or they're being used in a kind of a gray legal area where, in fact, there's no data being generated around them at all. And what we find is that's really putting people at risk. The example on the screen here is two women in the US who contracted HIV from a vampire facial. I don't know if anybody knows what a vampire facial is, but it's, to me, a really horrifying idea. But it's essentially injecting what you're being told are stem cells, your own stem cells, into the skin on your face, and it's intended to treat signs of aging, apparently. There's no evidence at all that it does anything except expose you to risk. And in this case, this is these women's own cells being put back into their own face, so there's no second person in here. So you could question, how did they end up HIV positive? Well, the facility that was actually performing this procedure was not meeting the types of standards of manufacturing that we would expect for a drug, and so they didn't sanitize their equipment appropriately. And their staff weren't trained on how to do that appropriately, and they didn't have to meet the types of standards we would expect because technically it's not a drug. So I think we need to be very careful around how we view some of these things and, not fall into the trap of thinking, well, it's your own cells going back into you, so it's probably fine, right? It's probably safe. That may not necessarily be the case. That said, some people are out of options. So if anybody in the room knows somebody suffering from a rare disease or a chronic illness and they've sort of run out of options, or you think about in the cancer space that people go through multiple lines of therapy and sometimes there isn't anything else for them. These are people who are looking for an answer. They're looking for another way to try and improve their quality of life or the longevity of their life. As a quick reminder, I have the hierarchy of safe ways to access drugs on here. Your first option always is Health Canada approved drugs. These have been through a rigorous review process. They've been through clinical trials. We know that they're manufactured in clean conditions. It's not going to be dangerous. Unfortunately, there aren't very many of these approved yet, as we've mentioned already. But there is a pipeline, and there is a really considerable amount of effort going into this space to try and bring more options to patients. The next sort of level down is accessing drug through a clinical trial. And there are many clinical trials in regenerative medicine ongoing in Canada and the world in general. Many of them are in rare diseases or in chronic illnesses where people don't have a lot of options, or in oncology where it's cancer treatment, the last line of therapy. This is something that physicians refer their patients into, so treating physicians will be part of some of these clinical trials. And then the very last resort option is to gain access through what's known as a special access program. And this is essentially a way to allow access to an unproven or unapproved therapy in Canada, not through a clinical trial. So again, this is led by physicians, and it's on an individual patient request, but there are ways to access these exciting new technologies, these innovations, for the people who need this. We just need to be very careful around how we structure that. Just to say that special access program is very similar to what you might know as a compassionate use program, or sort of last resort type programs, and they exist in many other countries. So the reason I say we need to be very careful with this is that there are other things going on in this space. Many of you will have heard about the right to try movement. This has been all over the press, particularly in the US in the last year or so. And I think we need to be very cautious when we talk about what actually is the right to try. So given what we've just talked about, and understanding that regulations are in place in order to protect public health and in order to structure and define what companies must do to show us that these products are safe, of high quality, and are going to be effective, we need to be careful not to accidentally dismantle a process that exists specifically to protect us. So right to try comes from a number of different places. There is a deregulation agenda behind it. There is also an access to medicines agenda behind it. There are many people who feel like they are excluded from the conversation and who don't feel like they have access to treatments that they think they should. And similarly, in the cell therapy space, there are many people who feel like, well, they're my cells and they're going back to me, so I should be the one to decide whether I get this or not and whether I can pay for it or not. But I think we need to be very careful and very cautious in those conversations to understand the true meaning of what those types of agendas actually lead to. We have a system in place in order to bring these types of therapies to patients and to develop them in a way that is going to lead to better health outcomes for all of us. We need to work with the system to bring these types of things to patients and to each other when we need it, rather than trying to dismantle a system and therefore have negative impact on all the other things that we do. So I'm going to leave it there because I think there's probably going to be time for lots of questions that way. Thank you very much. Hope you enjoyed it.
Fantastic, thank you. So we are ready now to kick off into our Q&A, but before we jump into the audience, there are a few important questions I want to make sure that we get through with our fantastic speakers. So if you all don't mind, I would love to hear, we talked a bit, just to make it super clear for everyone in the audience, what stem cell treatments do currently exist in hospitals? Let's start with that first, actually.
So I can talk from a Health Canada perspective. Health Canada has approved 3 cell therapies in Canada. One is, well, other than bone marrow transplant, I should say. One is Prochymal, which was many years ago and was actually the very first cell therapy in Canada. And then the next 2 are CAR T cell treatments, which are intended for the treatment of sort of end stage cancers. There's 2 different indications there. One is called Kymriah, and the other is, I forgotten the name now.
It'll be on the Health Canada website, easy to
Yes,
find. There you
yeah.
go.
So there's 3 of them that actually are approved.
Fantastic. And then I'm curious, what are are there any ongoing clinical trials that are exciting that come to mind? Like that you're most excited about, or you know that they're getting towards the later phases of the clinical trial spectrum?
In Canada, or?
Your favorite.
There's some cool stuff happening in the eye.
Cool.
Yeah, so I think this is in Japan. I'm not sure how far they are now, but a couple years ago is when I heard about they were starting, and it was starting to look promising. But they were starting to form these kind of sheets of retinal pigment epithelia, which actually we have an ophthalmologist in the office, maybe in the room. Maybe you can tell this better than I can. But the idea that you can kind of grow these sheets of cells that are supposed to be supportive for our photoreceptors. So people with macular degeneration, for example, start to go blind from the middle of their retina. Just starts to die off. And the idea is that this bottom layer of cells that is supposed to support and maintain our photoreceptors, which are the cells that see light, if we can establish them again, if they're healthy again, perhaps new photoreceptors can grow in the eye and we can start to see again. And so there was, kind of labs in Japan that were developing these very thin sheets of retinal pigment epithelium, RPE cells, that could then be implanted into the eye. And they had such a cool way of injecting it. It was it would like roll up, they would inject it into the eye, and then would kind of unfold and flap back into the back of your eye. So I'm hearing that that's kind of one of the exciting trials, but maybe you two have heard more about where it's at.
And that was one of the first ones. Well, in Japan, they have for the eye one of the first clinical trials actually using induced pluripotent stem cells. So it is happening. But like everything we talked about, like clinical trials are definitely happening. So a big one is in wound healing. So we're actually making sheets of skin out of stem cells, and being able to, if you have a burn, for instance, and actually put those sheets of skin on the burn, so it's actually a real piece of skin. So those kinds of things are moving into clinical trials, and that's really cool and exciting.
I heard they're also adding freckles and stuff to make it look more real.
Yeah.
It's true.
Yeah.
There's a lab that is developing a printer, and they're adding freckles to it.
I actually saw a talk recently about regrowing. If you get a skin graft because you've had some kind of skin damage, usually it's very difficult to get hair regrowing in that skin graft, and so there's some more research now trying to understand, because the same stem cells that produce skin also produce the hair in the skin, and so there was some neat stuff coming out where, at least in rats anyways, they could get hair regrowing from the graft, which is really nasty to looking little rat with a... Anyway, but very exciting clinically. And I would also say about the eye, we'll probably, hopefully, within the next decade or so, start seeing more stuff happening in the eye because it's a nice contained area. With that said, there have also been many reports of people who've had their eye sight worsened from undergoing the unproven types of therapies in the eye. So even though eye is exciting and we'll start seeing new stuff happening, I would still be careful. On that note, I would love to know, what are your top tips, experts, for spotting red flags when everyone here is excited and goes and Googles stem cells? What should they look out for?
I think my very, very first one would be if you're being asked to pay at any point, if you're paying out of your pocket. That's probably not a proven, effective, safe, high-quality cell therapy treatment. I say that because right now cell therapies are almost exclusively only administered in hospital, which means it comes through your public healthcare budget. So OHIP would be the one paying for it. So if you're seeing somebody and they're telling you that it's $10,000 out of pocket, it probably isn't a safe, or it isn't confirmed to be a safe and effective treatment.
What about those that are in the U.S., right? Like, people do pay out of pocket all the time for approved healthcare. How can we spot those frauds?
Yeah, I mean, there's still usually some pretty identifiable red flags. There's if for example there's no published literature on that product or on what they're doing. If you can't, really what you want to make sure you're doing is really a lot of research and reading around and checking that what somebody is telling you in a marketing brochure is in fact supported by data. So either you're in a clinical trial or they've run a clinical trial.
Yeah, because on that note, I have seen on a lot of these websites where they give data, they give papers, but these papers are usually like one mouse study that says, we did this and the mouse got better. That a mouse is not a human.
Yeah.
So the data has to be in real human patients. So a lot of them will say, look at all this science, you can click on all these links and it takes you to real scientific papers, real PubMed journals, but then you look at them and it was done in a mouse. It's not the same thing.
Or it's a case study of like one person in some other country was administered this and it looked like it worked, therefore you guys should pay for it today.
To counter that, you could be enrolling in a clinical trial where you are one of the first humans it's being tested on. But in that case, my red flag would be that would be receiving a ton of publicity. And so if you don't see that trial and the scientists leading the trial, that if they're trying to claim to you, well, you'll be the first, you'll be leading the way for other patients, and that's a very honorable thing that you might want to do, and a fantastic thing, but then it should be highly publicized in journals that you trust, and associated with schools that you've heard of before and that you that's kind of my rule of thumb. It's not perfect, but
You also shouldn't have to pay.
yeah.
If there's a price tag with it, it probably isn't a legit clinical trial.
And of course, the one therapy to rule them all.
That's what I was going to
Yeah.
say.
A lot of these stem cell websites you go on and the same injection treats everything from burns to Alzheimer's to autism to, there's absolutely no way, as Shifra
Erectile
mentioned.
dysfunction.
Yeah.
Yeah, the list will, if you click what can we treat and it's more
Longer
than one
than one thing.
it's a no.
Yeah.
Like as Shifra mentioned, approved trial, like something that's for Huntington's disease, cannot be used in Parkinson's disease, even if it may be beneficial for it until it's proven. So as soon as you see that list, just leave.
Especially for some diseases that we don't even understand. Like we don't fully understand how autism works. How are you telling me that this injection of stem cells into my leg or whatever is going to cure my autism?
Yeah. And it's pretty gnarly. I've heard of ones where they're trying to inject into your spine and then have the stem cells go up your spinal cord to the kid's brain. And this is happening in young kids, many of whom are nonverbal, to communicate the pain of all that. It's just bad news. I think there's a lot of other things we need to work on in our society accepting neurodivergence ahead of that. Wait, Nika, did we get yours?
Well, she said mine.
Okay.
Sorry.
So I put together a little checklist. It's not perfect, but these are some red flags I kind of use of how you can see if a study, any study really, but especially related to stem cells, is legitimate. And it mentions a lot of these things. I need to add the funding sources in relation. No, I need to add the cost thing. That's a good one to add. And then we've compiled a bunch of resources for you, actually. You could use the website. It's on my website, heysciencesam.com/stem cells, or you can use your phone's camera app. If you just open the camera app on your phone and point it towards this big code, it'll take you to the website directly. So this was just some way to point you to resources that are that many people like us would go to and that we trust and that many of our colleagues are putting out. So we're not specifically endorsing you to do all the things the websites, but they're good. There's good information on those websites to lead you in the right direction. Okay, so I think now we'd love to hear from all of you, and I'd specifically like to say that I like to foster a space of open questions. Don't be shy. If you're sitting there feeling like, oh, my question's probably silly or embarrassing, I don't know much about science or biology, those are the people we really want to hear from, because this is a space where we want to make sure that people can leave feeling a little more confident. So if you're a young student or you've never heard of stem cells before and this is your first time especially, we'd love to hear what kind of questions you have after this discussion. So we have some microphones circulating. Have some hands. I would love to take the first question from
Sam,
maybe some
I
of the
see
young people in the room.
Sam?
Yes?
There's a up top.
Sure.
What about xenotransplants? Like, say I wanted gills like a fish underwater. Would you be able to do that? And
That's
how
a fun
would I
question.
bypass the regulatory?
Okay. Well, this is. Can we use stem cells to become superhumans? Can I translate the question to that?
Not for yourself, unfortunately. There's currently no way to give you gills. However, we could potentially do that in an embryo of perhaps you want to be a father-son, treasure hunting underwater team, and you want to send your son underwater. So we could actually genetically alter the embryo. Now, I don't know about gills, but that's actually a huge, huge question in ethics right now. So as I told you guys, we can CRISPR, we can genetically cut up cells, the stem cells from your body, but that's not going to make a new person. But we can do this in an embryo. It's not impossible, whether that be stopping that baby from being able to get a certain disease, giving them a certain eye color or giving them gills. It's not outside of the realm of possibility. However, there's a huge international consortium coming together to debate if this is ethical, if this is safe, and what scenarios that would be okay. So I don't know if that answered your question.
But to the question of xenotransplantation, the idea of can we take like animal cells and put them in our
yes.
body, is that going to be okay? I mean, your biggest problem is going to be the immune system. Problem, not really problem. Like, it's there to protect us, right?
Yeah.
So if we were to take, fish cells, fish make gills, we don't make gills. We'd have to probably use the fish cells that make the gills and try to put them into our embryos or our bodies. You'd have to convince your cells to cooperate with those fish cells, and maybe part of that is cloaking the fish cells. I mean, I don't know. It's theoretically possible. You'd have to interface that gill with our respiratory system somehow, but perhaps the research of the future could head in that direction.
I think from the sort of regulatory perspective, like
She's like, oh God.
just gonna say currently that's illegal.
Currently, that's illegal. Yeah.
So there are very for anybody who's interested, there are very, very stringent regulations about what you can and can't do with embryos right now, and
Yes.
particularly in Canada, it's actually more stringent than it is in some other places. So you do see some kind of media hype from other countries where people are doing this and they're engineering embryos and then growing them up into humans who are different. That's currently illegal here. And there is, as Lise mentioned, a consortium of people coming together to talk about how do we do this in an ethical way. So there are many diseases that cause pain, that cause suffering, that we might be able to treat. We might actually be able to prevent this if we can edit the embryo rather than having to try and treat it when the person is already sort of out and about. So we need to consider whether the risk there is actually worth the benefit, whether there's definitely going to be benefit. If you were to somehow find a way to get implantable gills and it all hooked up to your respiratory system, you wouldn't be regulated as a person, but the procedure of putting that into you and connecting it all up would be, which is very similar to how we regulate surgeries, as an example. So, orthopedic surgeons replacing your knee, similar kind of idea.
It is a tough question. I would encourage everyone to think about and discuss even around the dinner table. Where do you draw the line in what types of gene edits and what types of cell therapies? When do you say it's okay to do, and is it only when there's a disease, or do we agree with enhancements? Those are really big discussions right now that we need more people to be a part of. So it's a great question worth considering. I would personally put my money more on cyborgs types of things happening sooner than a cell-based enhancement, because it just takes so long and it's so expensive.
I bet you didn't think your question would get that much discussion.
I mean, that's the stuff we like to think. It's fun. We have a question in the middle here. If we can use the mics is great just for accessibility.
There's one coming
Yep.
your way.
We'll wait. It's worth it. Thank you.
Is there any development in the development of synthetic stem cells? Are we looking
Synthetic in what sense?
Yes.
What do you mean synthetic?
well, simulating the real stem cell in a synthetic?
So we've literally from the ground up built the cell. Is that what you're asking? There are inroads in that field. So there are groups that are trying to understand how do we put together just the basic structure of a cell, membrane and the inside of a cell. And then how do we take the DNA, put that in there, and get the DNA to do what it's supposed to do, which is code for the cell's function. They're starting to make some interesting advancements, but not to a point where we can say we have synthetic stem cells, let's start making synthetic everything. But there certainly are researchers that are working and thinking just the way you are, that why don't we build the cell from the bottom up?
Mm-hmm.
Yeah.
There does seem to be things, though, that, biology, there's probably parts we'll never be able to solve, and that's, again, what's so magical about
It's
it.
true. But maybe we can do it better than nature. One can only dream.
Ooh, that's scary.
Here in the purple on this side.
So talking a little bit about along the lines of regulation and how we're a bit different here, there are some scientists, unfortunately, down in places like the States, that are trying to make chimeras between pigs and
Yes.
human stem cells in order to do something really good, which is to provide more organs for donation.
Yes.
What's your opinion of this?
Yes.
So, I mean, again, I will say it is a very heavily regulated space. So, those types of that type of trial, that type of experiment is regulated. You can't just go ahead and do it in your lab. So there is some oversight of it. And I think it comes back to an earlier question about how much oversight we should have and how stringent and how careful we should be. Typically at this point, a lot of these things are still being done from a sort of basic research perspective. Like what they're doing today is not going into a person today or tomorrow or even in a year or 5 years probably. A lot of it is really just about getting us more knowledge about what is it that matters in the development of that organ. Can we do without some parts of it? Can we cloak it in some way? Is there something that we can do? I think there's probably a lot of potential there, but it's something I'm quite pleased that we're navigating carefully because I think it will we need time.
I was just going to take a sec and I'm not sure if everyone's heard of this pig-human.
Yeah.
Has everyone heard of the pig-human chimera idea? There's a bunch of nods there.
Remind us.
Yeah, so basically the idea that, scientists came up with was like, we really suck at making 3D organs. I showed you those like beating balls. That's not a heart, right? So the idea was that the body does it better. So what if we use a pig as kind of like a living incubator that will construct our 3D organs and hold them for us until we need them? And the way that they thought they could do that was to take a pig embryo and they genetically modified it so that pig embryo could not produce a specific organ like a pancreas. So that pig could not produce its own pancreas. Then they took human embryonic stem cells and they inject them into the embryo. And the idea is that the human cells would then fill in the gap. Like the missing pancreas would then come from the human cells. So you would have a human pancreas in the pig growing. And it's great because the pig's size is similar, the organ sizes are very similar. There's a lot of similarities. So theoretically, you could just cut out that pancreas and put it into a human. The challenges there are, one thing is that we can't really control how many pig cells are then infiltrating the pancreas. Like the pancreas is not in a bubble, right? Like it's connected to the blood system, it's connected to nerves, it's connected to every other part of your body. So how do you stop pig cells from getting into this human-grown pancreas? The other thing was, I don't know if anyone saw the BBC's article, the pig with a human face one. Did anyone see that? They literally photoshopped a pig and molded a human's face into it. It was terrifying. But anyways, the idea there was that, we can't control where these human embryonic stem cells end up contributing. Like, we just stick them in there. They will fill in the gap of the pancreas, but they'll also go to the brain. They'll go to elsewhere in the body. And what if that pig then gains consciousness? Scary thought. This is like George Orwellian.
Yeah.
But anyways, there are, ethical questions around there. In terms of like personal stances, I think I'm a scientist. I like to dream about these weird ideas. I don't think we should necessarily push through to clinical trial. Let's try it. I think we do need to take a step back and see if we can understand what's happening there a little bit and maybe understanding how our stem cells integrate and how they then form this 3D organ within this vacuum of the pig's embryo could be interesting. So I think from a discovery perspective, it could be interesting. But whether we want to roll it out into a large clinical trial is a different question.
Fantastic. In the back?
I'm curious about stem cells being used for rehab, for regenerating like soft tissue damage, and a lot of that is happening in, I think, private clinics around the city and in the US as well. I'm not sure how regulated that is and how well that works and if people are paying out of pocket for it or not, so would love to hear your thoughts.
That's a great question. I actually have a map I didn't show, but there are, even in our city, not maybe 10 minutes away from where we are right now, there are unapproved stem cell clinics offering injections and I call them procedures, not therapies, because they're not therapeutic, really close by. So it's a great question. A lot of it is injury-related. Panel, what say you?
Well, I feel like this is for me. So yes, as I said, I mentioned that these are those types of therapies. All cell therapies are drugs in Canada, so they should be regulated as drugs. What has happened in the past is that some of these things have been performed as procedures under what's called sort of practice of medicine. And if there's minimal manipulation of cells and therefore they're being taken out of you and put back in for the same, to do the same thing they were originally intended to do and they haven't been altered and so on, there is a little bit of space in the regulations that allow physicians to do some things in practice, in their own practice, and that's actually regulated by the College of Physicians at a provincial level, which is why some of these things have kind of started. Unfortunately, what's happened is they've sort of escalated and exploded to this place where we now have, as Sam said, dozens of unapproved therapies, which should be regulated as drugs, and in fact are drugs under our regulations, but are already out there and already being given to people, and people are paying out of pocket for. The real problem there is actually we haven't yet seen a lot of sort of safety issues with them. We haven't yet seen people who've lost a leg because this was performed incorrectly in their knee or whatever. But what it does actually is it pushes the science back. It stalls us at a point a few years ago where, in the last 5 years, we could have been collecting data on these procedures and actually proving that they work, and then getting them to a point where we can definitely help people with it. And instead, what's happened is this data is lost because individuals are just going, to their local physio or whoever it is that's performing these procedures, and that person is not collecting data and is not, analyzing it in the same way. So we can't even say for sure that the people who were treated before actually did well or weren't harmed. So we're still at zero. We still have nothing on these procedures. And it is something that Health Canada have started to try and crack down on, same as FDA in the US. And you will have seen some press around that sort of in the summer this year, I think. There were some sort of cease and desist letters sent out. But it is an ongoing issue. We haven't solved for it yet.
And as a devil's advocate, I mean, it's not a perfect system, and both patients and doctors get frustrated at how long it takes, and that's a fair problem to have. And if you think it's relatively safe, you may go for it. And as an athlete, I get offered a lot of these different kinds of injections, and that there may be anecdotal evidence that they work or that they help, but and usually the doctors are very open that I can't actually guarantee this is going to work, but it's an option. So I mean, that's where you can kind of run into that issue of trying to decide for yourself if that's something you want to do. But hopefully Shifra's explained to you why it's actually not a good idea to move forward with those kinds of things.
Yeah, I mean, sometimes placebo can be powerful, right? Like those studies have been shown. And if the placebo is not doing harm, then it's an individual's decision. Do they want to go ahead with this unapproved treatment? And I fully empathize. I understand the position that some people are in, and they're really, desperate, and they want to try. But on the other hand, I think we have to look at the big picture too. We have a responsibility to recognize that there's risk. So if something does go wrong, let's say they took embryonic stem cells and they injected it in my knee, that would not go well. That would end up, forming a teratoma. I'd have like a ball of...
A tumor.
Yeah, a tumor. It would be a ball. If you cut it open, there'd be like bone and teeth, and it's not pretty. And imagine if that story got out. That would put the field back. And that's happened before, right? So these sort of horror stories of unapproved therapies getting used, if they do harm, then everyone's going to get afraid. They're going to be like, well, stem cells are horrible. We should not invest any money there because look what they did, right? Yeah. But it's not giving them a fair chance. So that's kind of...
Yeah, that's why we get so scared, because we're worried about the patients, and we're also worried about the perception and what that will do to the field that does have promise but just needs time.
Sorry, I was just going to mention as well that sort of the placebo effect piece. I mean, the other part, of course, is that you are paying out of pocket for these types of treatments. So if you've spent $10,000 on something, subconsciously you're going to be looking for whether you got any return. So even if there was no effect at all, your mind is still going to be grasping at something that made you say, oh, actually, maybe I do feel a little bit better. Maybe that 10 grand was worth it. Maybe I should do it again. And I think it's very, very difficult for us to decipher what actually is an effect and what is just... the placebo effect, the sort of the conditions that you're in, and the how the level of sort of desperation that people are at as well.
Which is why clinical trials are so important.
The takeaway message. I think we have time for a few more questions.
Sam, there's one up at the very top here.
I'm sorry. Yes.
If you found a way of growing skin cells that will produce hair, you've got a treatment for baldness. Could it cause the funding for the field to explode?
Yes.
Very true.
I have two questions about the related fields. One is cancer research, and the other is the ability of amphibians to regrow
Yep.
legs and so on. Has there been any synergy in the research between those fields?
Great question. So I think we're looking here, how do we look at other animals for inspiration in regenerative medicine, including amphibians who can regrow whole limbs, et cetera, and then applications to cancer research, I think. Fun one.
Yeah, I mean, so when you go to scientific conferences, and I've been to a few, there's sessions of different types of researchers working in different areas of regenerative medicine. Those that are working in the very sort of basic science of understanding how an axolotl, for example, would regrow its limb when you chop it. Their progress has been great, and we can understand a lot more about the genetics that goes into like limb reconstruction. But are we at a point where we can take those ideas and see if we can get that to work in a human? No, I would say, because we are really at the reverse engineering step. We're reverse engineering how that organism works before we can see if we can convince our cells to mimic that. I think it would be possible to regrow a human limb. Like, our stem cells are able to do it, they're just slow. So you'd have to come up with some sort of approach that would really speed them up. But I mean, this is speculation. I like to dream, as I said, as a scientist.
I wouldn't mind commenting on your question about the cancer therapies. So what Schiffer mentioned is actually one of the approved therapies is against cancer. And if you remember the slide I showed where we're trying to hide our cells from the immune system so those T cells can seek out things that are not supposed to be in our body, well, what scientists have done is gone into the genetics of those T cells and they've trained them to seek out tumors. So they've trained them to note that cancer is bad and we don't want that. And then you put those T cells back in your body, and when they find cancer, they destroy it. So that, like I said, we said is an approved therapy that you can actually get. And so the advances there are that they're now trying, like that's for blood cancer, so it's dispersed in the blood. They're trying to train it to find tumors. And on the personalized medicine front, everyone's tumor is different. So I mean, they're now taking a biopsy of your tumor, sequencing that, putting that sequence and training the cells that that particular sequence for yourself is bad. So I mean, the field in that way is just exploding. And I think those therapies are going to be the ones that are coming out the quickest, since we already know how to get them through the approval process. And it's super promising.
And I would say also that really at this point, probably 50% of the research in RegenMed is in cancer research. Like there is a huge focus there. So there's certainly, people are trying to find applications to sort of cross-pollinate.
I think
Sam,
we
there
have
was-
someone with a mic already here.
I was
Yes.
going to say there's also one down here who keeps
Okay.
putting his hand up.
I know. Well, we'll have refreshments after, and we'll be wandering around. So you'll have time maybe to ask one-on-one as well. But yes.
So all of this research has been in regards to stem cell application in humans. But I was wondering if there's any research or interest into research for stem cells about animals and therapy for other animals, like in a veterinary sense or ecological and environmental sense.
Yes,
Yeah.
I know of. I know of two off the top of my head, and these are people who came in with the intention to study human stem cells, and they were like, "Well, why don't I do this in like racehorses?" I mean, first of all, there's a lot of money to be made there, but also there's a lot less regulation, right? So those people can move forward with their translational research less hindered by regulations. I mean, I say hindered in a negative sense. It's really not negative, but to a scientist, it can feel, I guess, somewhat suffocating when you really want to try your stem cell therapy, but you can't. But you could do that in like a horse or a dog more quickly. So there are researchers who are doing just that, and it's not secluded, right? Like if we learn something interesting in a horse or a dog, maybe that can inspire something interesting to happen in human studies more quickly. So they're synergistic in that sense.
Yeah, I mean, we have a manufacturing facility in downtown Toronto now, and we're not allowed to do animal research, but we've been asked to manufacture stem cell therapies for animals. So, I mean, it's definitely already moving in that direction.
And I was just going to point out as well that like one of the really key additional regulatory considerations with animals is actually about how those cells might end up in our food chain, because so many people eat animals. We now need to be careful also that if we're making changes to the DNA of a pig and then somebody eats that pig later on, is that going to affect us in some other way? So there's sort of different regulatory considerations there that aren't quite the same as we might need to worry about with humans, but there's definitely issues also.
I hadn't thought of that. That's fascinating. In the corner up top, yes.
Okay, my question is, how is stem cell research being used
Great question. So spinal cord therapies. Anyone want to
You know
talk
a lot
about
about this. Do you
it?
want to?
I'll let you. I'll go. Anyone? No.
I mean, I've heard of some trials that had started. There was the Geron trial that was aiming to treat people who had lost function of their limbs, so paraplegics or quadriplegics, I can't remember. But so their idea was to take human embryonic stem cells and derive these not fully mature neural cells that they could inject into the spinal cord and hope that those cells would support the healing of the person's spinal cord. And that trial got suspended, but I don't think it was for adverse reasons. I think they decided it was expensive and they wanted to move the funding elsewhere. In terms of actually approved, to my knowledge, that's not approved anywhere.
No.
And if
No.
someone's claiming to treat your spinal cord in China or Singapore, I would be highly skeptical. But perhaps
No, you're right. It's not approved anywhere,
Yeah.
but there are definitely clinical trials ongoing all over the world, some in Canada and obviously other places. There are also stories of many not so reputable outfits offering this as a treatment, as a therapy, which you would pay for. And there have been anecdotal stories about side effects and adverse events there, which are pretty serious. So, it comes back to the checklist that Sam had before, that, think carefully about what's being offered.
There is a really, one of the ones I've been following excitedly over the last few years is coming out of the Bay Area in California. It's the Asterius trial. And that one has had some exciting early results. But if you look up Asterius, I can help you find it later. You'll see how kind of long it's been, and you just need to be extra careful dealing with the central nervous system, especially the spinal cord, because it's a very small space, and adding stuff in could go awry quite quickly, and the connection to the brain. So
the short answer is there's clinical trials everywhere, not approved anywhere. But
and super promising research in the field as well, which is why you hear a lot about it, because it probably is very, very promising. It's just not there yet.
Yeah.
Okay, I think with that, we're heading in. It's 3:30 now, so I'd love for everyone to join us for some refreshments outside, and we can mingle and chat more. I want to thank you all so much for coming and having this exciting conversation with us. And of course, if you could join me in giving a round of applause for the organizers and our wonderful panelists especially. Thank you.